Hello, my name is Ewan. To give you a bit of context about why I'm giving this talk, as I was growing up, I was fascinated by dinosaurs. So naturally, I watched Jurassic Park. Many of you have probably seen it too, or Godzilla, or some other movie with genetic modification. So when we think of genetic modification, these are our first thoughts. It is something scary, and it involves monsters with big teeth, eating people. By this point, you're probably wondering where I'm going with this talk. After all, most of us don't work in biotechnology labs or visit islands with questionable containment measures for large reptiles. However, the reality of genetic modification is quite different to Jurassic Park. The first genetically modified organism, or GMO, that consumers interacted with was used to produce insulin to treat diabetes. This isn't a new development either. Genetically modified E. coli was FDA approved in 1982. The FDA also approved genetically modified crops in 1994, when nutritionally enhanced genetically modified tomatoes were found to be as safe as the traditional varieties. So, unlike Jurassic Park, genetic modification is not the story of humans against nature, but it is instead humans working with nature to solve our problems. This makes it vitally important to understand how genetic engineering really works and how it can be used, because we're currently facing a lot of problems that gene editing can help us solve. Now, I want you to imagine your favorite sports team. During a game, one of the players might get injured and need to be substituted off the field. CRISPR, which is a natural gene editing mechanism in bacteria, works in a similar way. It targets the player, or nucleotide, that needs replacing and swaps it for another member of the team. On a football team, there are 11 players, with another 5 substitutes. But let's imagine we want to change one player in a team of over 6.4 billion, or roughly the world's population in the early 2000s. To help CRISPR make the right substitutions, a PAM sequence is used to locate the target gene. You could think of this as the unique number on the player's shirt. Once the correct PAM sequence has been located, the cargo DNA carried by the Cas9 enzyme can be inserted into the genome. In this way, CRISPR acts as a pair of molecular scissors, cutting and pasting relevant genes from one organism to another. To give you a current example of this, quite recently, the FDA approved the farming of genetically modified pigs. One of the largest problems facing the pork industry is porcine reproductive and respiratory syndrome, or PRRS for short. This is a viral infection that weakens the immune system, causing miscarriages and higher infant mortality. In the US alone, over $1.2 billion are spent every year on controlling the spread of this virus. Why? To give you a sense of scale, an outbreak in 2006 in China infected over 2 million pigs and killed over 400,000. The genetic modification in this case is relatively minor, equivalent to changing 3 players in a team of thousands that work to produce a protein that breaks down damaged red blood cells. This tiny modification is enough to stop the virus from infecting the pigs, while retaining the original function of the protein. Additionally, this mutation is likely to have occurred in nature at some point in time. So instead of reinventing the pig, this just distributes this protection throughout the population. Another instance of CRISPR being used to combat infectious diseases is infectious diseases with an insect vector, such as dengue fever. Dengue fever kills over 36,000 people per year. And in order for the mosquito that transmits the virus to infect people, it needs to be continually reinfected by blood that carries the virus. So, one way to prevent dengue would be to stop the mosquitoes themselves becoming infected. Luckily, mosquitoes already have 3 pathways to control viral infections. So, increasing the expression of the genes AGO2, DCR2, or R2D2, sorry, not that R2D2, would be a way to control the spread of dengue. You could think of this as changing the amount of time players spend in a particular part of the field, rather than the players themselves. Moving on to a human context in which CRISPR could be applied would be treating genetic conditions such as sickle cell anemia. Sickle cell anemia is caused by a single nucleotide mutation in the gene responsible for producing hemoglobin, which is the protein that carries oxygen throughout our bloodstreams. This mutation makes red blood cells less efficient, reducing blood oxygen content and causing increased pain, swelling, and fatigue, alongside other symptoms such as a weakened immune system. Because of the simplicity of this mutation, it will be relatively easy to correct using CRISPR, changing one player in a team of billions to make a huge difference to a person's everyday life. That sounds good, right? It should. However, people who have only one copy of this mutated gene and one copy of the regular hemoglobin-producing gene suffer almost no negative side effects, while being more resistant to malaria. This is a huge advantage in sub-Saharan Africa, which explains the high prevalence of sickle cell anemia in this region. Now, I would argue that with modern medicine, especially treatments for malaria, using gene therapy to remove this mutation would not overly impact those people who have one copy of each gene. However, this example demonstrates that even when correcting a harmful mutation using CRISPR, there may be unintended consequences. Okay, all of this progress doesn't mean that CRISPR is always used as a force for good. He Juan-Qi was imprisoned for 3 years following his illegal experimentation on human embryos, where he disabled the CCR5 gene in order to make them immune to HIV. However, at the end of the day, CRISPR is just a tool, a method of changing the dynamics of a genome through substitutions. We humans are the managers for this tool, and we decide which players stay on the field and which get substituted off. Time will tell if we get the game plan right. Thank you.